And our next speaker is Casañal, who's going to tell us about, oh, it's up there. I haven't got my glasses on my face. A hub for 3' end processing, structural insights into mRNA polyadenylation.
Hello, everyone. First of all, I would like to thank the organizers for the nice organization and for the invitation, and to present my work here today. So I am interested in understanding how complexes involved in RNA processing work. So after DNA is transcribed into RNA, the nascent RNA undergoes different types of processing, including 5' capping, splicing, and 3' end processing. Once the RNA is modified, then it can be transported from the nucleus to the cytoplasm for its correct translation by the ribosome. In the 3' end processing machinery, there is one complex that is called cleavage and polyadenylation factor, CPF. This is in yeast. In human, it's called CPSF, which main activity is to cleave that nascent RNA and then add the poly-A tail. And the poly-A tail is important because it confers stability to the nascent RNA. It promotes its export from the nucleus to the cytoplasm, but it's also important to enhance translation. So the length and the position of the poly-A tails is also important because this is going to have an important impact in gene expression regulation. And we also know that defects in this machinery is involved in different types of diseases, like including cancer, some neurological disorders, but also blood diseases like beta thalassemia. And in this case, for example, one single mutation in the poly-A signal, that it's where it's a specific motif in RNA that is going to be essential for the addition of the poly-A tail, one single mutation is the cause of beta thalassemia. So CPF, the cleavage and polyadenylation factor, is a complex of approximately 1 megadalton that is composed by 15 different subunits. It's essential for cell viability, and despite its fundamental importance, there was still not structural information about it. It presents 3 main enzymatic activities: the endonuclease that cleaves the RNA, the poly-A polymerase that adds the poly-A tail, and 2 protein phosphatases that dephosphorylate the C-terminal domain of Pol2 and coordinate transcription. So, however, there are still many questions that need to be answered regarding how are the different enzymatic activities of the complex coordinated, and how do they regulate the processing of RNA. So, this is an ideal target for cryo-EM. As you can imagine, it's a 1 megadalton complex. So when I joined Lori's lab, there was not structural information available about it. So the first thing I did, it was improving the purifications of the complex. So when I first explored this complex on cryo-EM grids, both negative stain and also in cryo, the protein was always falling apart. So then I had to find strategies to stabilize the complex on the grids. And what I did is doing cross-linking of the sample. So I used this specific cross-linker, BS3. And here we can see a gel where we see that cross-linking reaction. So this is a time course experiment in which I mix our protein with certain amounts of the cross-linker, and then we could see that after a certain amount of incubation time, then we can see that the complex was actually cross-linked. When we do cross-linking, one of the main problems we have is that this can affect the conformation of the protein, and it can force the protein to aggregate. So it is important to verify that actually the protein is still good. So this was the first image that we obtained about CPF. And actually, these were very exciting times for us. But you will agree with me that even if we could see some individual particles there, some single particles, this still was looking quite messy, actually. And when we obtained our 2D classes, actually what we were seeing is that the sample was highly heterogeneous. And we can see here that we have 2D classes with different sizes. So when we did our first reconstruction, what happened is that we obtained a map to a resolution of approximately 12 angstrom, but the first thing that this was telling us is that this was too small to be the full complex. For the size of 1 megadalton complex, 110 angstrom complex was too small. So this meant that the protein, even after cross-linking, was still falling apart when we were making the grids. So we had to think of a different approach to study this complex, and then what we were doing at that time was a collaboration with Karen Robinson to do native mass spec for the complex. And native mass spec has been proved to be very important to determine the composition and stoichiometry of macromolecular complexes. So what we did is that we took our native complex and we flew the complex into the mass spectrometer, and the ionization caused fragmentation of that complex, and then what we were able to detect was 38 different complexes that after analysis, computational analysis of them, allow us to determine a new network of protein-protein interactions for the complex. So we defined then a new architecture for CPF, and this was actually very interesting for us to see because the three main activities of the complex, the nuclease, the polymerase, and the protein phosphatases, were segregated into three independent subcomplexes that now we call the nuclease module, the phosphatase module, and the poly-A polymerase module. So then we decided to focus on the polymerase module because this is the one that contains the poly-A polymerase that it's going to add the poly-A tail. So this subcomplex is formed by 5 different subunits. CFT1, which is a large subunit of 150 kilodalton, which all we knew at that time is that it had a high contact in beta strands. YTH1 protein with 5 zinc finger domains. PFS2 protein formed with a WD40 domain. FIB1 small protein with intrinsically disorder. And PAB1, which was the polymerase. So the kind of questions that we wanted to understand here were like, okay, so we know that the polymerase is capable to polyadenylate on its own. So what is the role of the different subunits in the complex? Why are they necessary? How are they assembled? So at this point, we decided to purify the complex recombinantly by using the baculovirus expression system and using insect cells. And we obtained the complex in the presence and in the absence of the polymerase. And then we reconstituted the in vitro activity of it. So here what we see is a polyadenylation assay in what for a given RNA, we see that in the presence of the complex and ATP, we can see that that RNA increases its size because polyadenylation is happening. So this is a typical micrograph of our complex. So I have to say that when we were imaging the complex in the presence of the polymerase, this was still quite heterogeneous, so we decided to go for the sample that didn't have the polymerase because it was behaving better on the grids. Now we didn't need any cross-linking, and this is how our images were looking like. And you will agree with me that now this was much nicer than before. And when we look at our 2D classes, the internal details that we could observe were much higher. And also here, I think you could recognize as well this specific view of the complex. So one of the main challenges that we face nowadays in cryo-EM is sample preparation. So when we collected our first data set on this complex, we observed that actually our protein had, or our complex had preferred orientation. And we can see that here. And this was translated in the fact that we didn't manage to increase the resolution as much as we needed to do a de novo building reconstruction. So what we then tried was to collect to mobilize the complex into different supports. We tried carbon, we tried graphene oxide, but we also use different ice thicknesses. And this was, in the end, what we needed is to collect quite a large amount of data. So we collected data in diamond and also at the LMB. And what we observed is that by a combination of all these factors, we managed to obtain enough views to obtain a reconstruction with a resolution high enough. So this is just to show you that we were working in diamond when the microscope was still at the beam lines. And well, this is at the LMB. And at the LMB, it's very common to work as teams. And this is my colleague Anand that I'll show you a bit later, a picture of him. So because we needed to collect data in different microscopes, then the next challenge we had to face is how to merge that data. So, and the way we did that was by obtaining a high-resolution reconstruction, or like the highest resolution possible, from each data set, collected at different microscope. And here we have, like, this was our reconstruction from the diamonds data set, and then from our cryos2 at the LMB. And then once we do that reconstruction, what we have to do is to superpose them to find what correlation there is between them so that we can normalize the pixel size of one to the other. So with this strategy, we managed to obtain a map to 3.6 Armstrong resolution, and this was like, this really made a difference for us. So we were moving at a resolution of 4 Armstrongs, and at that resolution we didn't see separation of beta strands, but once we were to 3.6 Armstrong resolution, we could see that, which was important, again, for the model building. So we did classification to classify heterogeneity using Reliant, and we obtained one specific class with 77,000 particles to a resolution of 3.5 Armstrong. And another one that was showing a different conformation, but we thought it was a different conformation at that time. So when we superposed those two classes, those two reconstructions, what we saw actually is that we didn't have change in the conformation, but what it was happening is that in one of them, we were missing part of the complex. And we can see that the rest was staying basically the same. So if we go one step back, and then we look again at what we were having with the full complex when we were purifying the full complex, was this reconstruction. But if we compare them both with what we were having now, actually, the 2D classes were very similar, and also the complex. So we were coming to the same result by the two different methods. But with the difference that now we were obtaining a very high resolution structure in comparison to before, when it was still much more heterogeneous. This is an example, or how to show the local resolution in our map. The local resolution is quite homogeneous, and then you can see that actually here we have some beautiful alpha helixes and how we can see the separation of the beta strands. So the next challenge was then to do de novo building. Why? Because we didn't have any crystal structures that we could fit in our model, so we had to face that problem from scratch. This is an example of how our density was looking. Our map was looking for the alpha helix, beta strand, and some of the zinc finger domains that we have in our structure. And at that time, Paul and I started to work together. And I was actually very fortunate to be involved with him in the developing of cryo-EM code for cryo-EM. So here, it's how our map was looking like. We see here one of our beta propellers in CFT1, and then we can see that some of the side chains at this resolution are still not very well defined. So some others are very well seen, like this one that we have here, but especially aspartic acids and glutamic acids, we in general don't see them very well. And this is what Paul was telling us yesterday. So for model building, Paul had to introduce a number of different algorithms so that we could fit low resolution, we could fit models into low resolution maps, because the fitting of even bulky amino acids is not going to be the same. So here is our result. So here we can see 3 of the 4 components that it's forming our protein. So we see in green CFT1, the one that presents a huge amount of beta strands. So it's composed of 3 beta propellers, 1 of each side, and then 1 at the bottom, and then a WD40 domain, which is BFS2 sitting on the top. And then on the left, what we see is YTH1, the protein with the zinc fingers. So how is YTH1 in pink interacting with the complex? So we see that it's formed, we know it's formed by 5 zinc finger domains, but what we see is 2 of them. So they have a coordination with 3 cysteines and 1 histidine. And we will see that this is interesting, actually, because the rest are going to be flexible. And then the protein wraps around the side of CFT1, and it goes inside, it's inserted in the center of one of the beta propellers. So BFS2 in yellow is sitting on the top, and it's interesting to see how this interacts through an extended alpha helical region that it's inserted into this cavity, varying a surface of more than 4,000 square angstroms. And this is a very tight interaction. And this was important for us to see, because in the past, we were never able to purify these components independently. So they really need the presence of each other so that they can be correctly folded. So at this point, it was striking, actually, to see that the fold of this machinery is also conserved in other machineries that are important for nucleic acid binding. So for example, we have here the example of DDB1, DDB2, which is involved in DNA repair, and it's capable to bind DNA through the DDB2 protein, which is sitting on the top, similar to what's happening in the poly-A polymerase module of CPF. And then here, another example is the SF3B protein of the pre-splicing machinery that is capable to bind also RNA. So because our machine is going to be processing RNA, we propose, but after analyzing our structure, that this was going to be a potential putative RNA binding surface. So if we explore in detail what is happening on the top, we can see that that part is very rich in arginines and lysines, and that's going to be likely to be an area that it's going to be binding RNA. But also, the zinc fingers are important, and we know that biochemistry that are going to be binding RNA. And actually, recently has been published the human structure of these same homologous, and they have obtained the structure in the presence of the poly-A signal of the poly-A signal, which is this motif of RNA, and they have seen that it's actually bound to this part of the complex. So, as I said before, we are seeing here 3 of the 4 components that we have in our complex. So why is that? So the components that we don't see there, they are flexible, and we don't see them in the map. So we had indications from biochemistry assays that that these parts were going to be flexible, and then we decided to do cross-linking mass spec to see what was happening there. And what we observed is that actually this zinc finger protein, it's binding through its C-terminus to FIB1, which is a small protein that is very disordered, and that FIB1 is the one that is actually bringing together the polymerase to the rest of the complex. So this was making sense for us, because also the polymerase has to be tethered flexibly to the rest of the complex so that it can bind different types of RNAs and then possibly bring them to the right position when they have to be cleaved and then polyadenylated. So this was part of the cross-linking mass spec experiments that we were seeing, and then this is how we saw like the different connections between the parts of the complex. The 3'M processing machinery also has other factors other than CPF. So one of them is the cleavage factor, CF1A, but also we have to remember that this subcomplex has to bring together the rest of the components of CPF and also RNA so that it can be processed. So we were able to determine that this factor composed of 4 different proteins is actually activating or stimulating the activity of the poly-A polymerase module of CPF. And first, I show you that we were able to identify that they were interacting. For that, we did pull-down assays. And what we saw is like here, in the first lane, we have our complex that then when we mobilize it to some resin of beads, and then we pass our other protein, they were binding. And then we could see that specifically it was binding to the subunits RNA14 and RNA15 of CF1A, which are these ones here, which we know are important to anchor the RNA in a specific position. And here is how we can see that they were important. This interaction is important for stimulating the polyadenylation activity, because if we compare in a polyadenylation assay what happens in the absence of CF1A and in the presence of CF1A, this reaction is happening much faster. So just to summarize, I've shown you the structure of the poly-A polymerase module of CPF, which is composed of CFT1, PFS2, and YTH1. There are some flexible parts that we don't see in our structure, but that they are going to be important to bring together the polymerase to the rest of the complex. And that this complex is capable to bind other accessory factors that are essential to anchor the RNA to the machinery so that it can be polyadenylated efficiently. But also we have to keep in mind that this is going to be the core of the complex that it's going to bring together the rest of the components of CPF. And with this, I would like to finish with some acknowledgments. Specifically, I would like to thank Laurie Passmore, because she's been very important to encourage us through this challenging project, and then my colleagues, Anand and Chris, with whom I've been sharing this project from the beginning, and of course, the people at the LMB that have contributed very actively to this work, specifically Paul Emsley. And thank you for your attention.
Questions for Anna? So I'll start you off with a difficult question, I think. So you paint a picture of having quite a flexible linker between your zinc finger domain and the polymerase. Do you think that precludes the structures of larger complexes, or are there enough interactions
we think that acts actually as a platform. It's like to bring together different pieces. And we have been now able to identify exactly what part of the zinc finger domains are going to be binding that specific protein, and also which ones, like the fragment that is bringing together the polymerase. So we hope that we can now stabilize the complex further.
In your complex, you have one of the components is intrinsically disordered. Mm-hmm. Its presence result in a does it make the maps worse?
Does it make the?
Make the EM maps worse? Did you try to exclude it from the complex and try to acquire data without it?
So we are not working on that, on having like a smaller complex, but also larger complex, because we think that that's going to be stabilized by other components of the machinery, but we don't see it at all. it's not, in our 2D classes or in our micrographs, we just don't see density at all for that. But it's very stable in solution, so.
I'm curious about your initial attempts to purify the whole thing, and then you realized you just saw the polyadenylation complex. Have you tried to purify the other two modules by themselves? Are they just disordered, or is it when you try to put them all together that?
So we are working on that, and now we have like, we are also studying like them independently and also like them binding to the core part of the complex, and there are other parts that are also flexible. And also if we study, we analyze the domain organization of some of them, we actually see that in many of them. But this might be again important for RNA binding interactions or for protein-protein interactions. So we have to find ways to move forward for that.
I was wondering, how confident are you in the chain tracing and at that resolution? And can you use the systematic absence of certain side chains, glutamates, aspartates, and so on, to validate chain tracing?
So I'm confident in the sequence assignment. What it was challenging in the beginning was I was used to build models for higher resolution, and then moving into 3.5 to 4 amps of resolution, that was a challenge because I wasn't used to identify side chains at that resolution. But once you start working with it, you can really see the difference between them. But then you will be analyzing more like you have a large residue, then small, medium, and then you have to work in a different manner. There are also some programs like ProSmart that it's also included in CUT that help for the sequence assignment. For the second part of your question, when you were saying, you were asking if the absence of some of them can help you, I don't think that's the case.
In what way does ProSmart help with sequence assignment?
So you can take homology models and then you can place them into your, so actually the way we were working with this structure was by creating some specific homology models. The homology models where the sequence identity was very low, so it was in the range of 15%, but then you can still bring them in. You are gonna always have some regions that are gonna be conserved, and then you can start assigning from some specific areas that are gonna give you information, and from there you build up. And then, for example, in the core of our complex, we had a very well-defined helical fragment that was, with no doubt, it was very easy to assign. And then from there, again, we could start like building up for the rest of the complex.
Okay, I think we'll, in the interest of time, we're smack on time, so we'll leave it there. But please join me in thanking both speakers for a pair of wonderful talks.